Good morning, students. We are continuing the topic mutation. We have done mutations were first of all discovered by Hugo de Vries. Mutations, they are divided into two categories, chromosomal and gene mutations. We are finished yesterday with chromosomal mutation, which were of two types: change in the structure of chromosome and change in the number of chromosomes. Change in structure of chromosome can be brought about by deletion, inversion, addition, and translocation, whereas change in the number of chromosome is brought about by two processes, aneuploidy and polyploidy. We have done what is the cause of aneuploidy. It was non-disjunction, that is failure of separation of homologous pair of chromosomes during gamete formation, and in this category what arised, that is monosomy, trisomy, tetrasomy, double monosomy, double trisomy, and nullisomy. Now, second type of mutation due to change in number of chromosome is polyploidy. In aneuploidy, there was addition or deletion of one or more chromosomes, but in polyploidy, the whole set of chromosomes is duplicated and it results in the formation of triploid, tetraploid, pentaploid, and hexaploid individuals. Organisms, such organisms, they are sexually sterile, and because during gamete formation the synapse formation does not take place. And this polyploidy, it can be induced by phytohormones or certain chemicals like colchicine, colchicine. Now polyploidy is further of two types, that is autopolyploidy and allopolyploidy. In autopolyploidy, the same genome is duplicated and it leads to Gija's effect or giant effect. It is applied in maize, rice, and gram. Like you have seen these days, fruits, they are bigger than the normal size. The size of grapes, guava, and apple, it has increased than the normal size. It is all because of the polyploidy. The polyploidy is induced with the help of phytohormones like auxin, gibberellins, and these hormone fruits are treated with the phytohormones, and this results in polyploidy. Means the such fruits, they are either triploid, tetraploid, or pentaploid, and then and they are sterile, that is why they are seedless also. So this is the result of autopolyploidy. And second is allopolyploidy. It is developed through hybridization between two species followed by doubling of chromosomes. For example, wheat, tobacco, and cotton. Wheat, which we eat, is a hexaploid, and it has two types of genomes. Four genomes means four set of chromosomes of one breed and two set of chromosomes of another breed. So if different types of genomes, they are duplicated in the same individual, this is called allopolyploidy, and if the similar genome is duplicated, this is called autopolyploidy. Now the second type of mutations, they are the gene mutations. The stable changes in the genes, they are called gene mutations. The change in one or few nitrogenous bases, uh, these changes are very minor and they cannot be observed by microscope. Gene mutation हो गई है, it can be detected only when it produces the altered phenotype. And a nitrogenous base, single nitrogenous base, which is capable of undergoing mutation, that single nitrogenous base is called muton, and in a gene there are present several mutons. Now what is mutagen? Any extracellular physical or chemical factor which is able to cause mutations is called mutagen. Mutagens, they are of two types: physical mutagens and chemical mutagens. Physical mutagens, they include temperature and high energy radiations. Increase in temperature by 10 degrees Celsius, that is, increase in temperature by 10 degrees Celsius, it increases the chance of mutations up to 5 times. Means जैसे जैसे temperature बढ़ेगा, for every 10 degree rise in temperature, chances of mutation increase by 5 times. And high energy radiations like alpha particles, gamma, beta, x-rays, ultraviolet rays, cosmic radiations, these, they also cause mutations because these radiations, they ionize DNA, means they convert DNA particles into ions, and you know ions, they possess more energy than the neutral atom. And the nuclear bombs, they released the radiations, and this led to many mutations in the individuals. Second category of mutagens is chemical mutagens. Chemical mutagen examples are, the first mutagen is nitrogenous acid, not sorry nitrogenous, it is nitrous acid, nitrous. Now presence of nitrous acid, it leads to, it changes cytosine to uracil, and it causes pairing of guanine with xanthin, xanthin, and it causes pairing of adenine with hypoxanthin, hypoxanthin. Now nitrous acid, it causes mispairing, that is, normally A pairs up with T. Now adenine, now it is replaced by hypoxanthin, and T will pair up with cytosine. So this AT pair is replaced by HC, and normal CG pair, C cytosine is replaced by uracil and guanine is replaced by xanthin, so CG becomes U and X. So this disrupts the replication and transcription, and this leads to incomplete or defective polypeptide. Second chemical mutagen is base analog. What are base analogs? They resemble normal nitrogenous bases of DNA, and they get incorporated into DNA in place of them. For example, 5-bromouracil and 5-fluorouracil, they substitute T and pair up with guanine. You can see guanine is replaced by bromouracil, and guanine, it can be replaced by fluorouracil also. The third chemical mutagen is nitrogen mustard. Nitrogen mustard is a gas which acts as alkylating agent. Alkylating agent means we know that two polypeptide chains in DNA, they are joined by hydrogen bonds. So now this instead of hydrogen bond, two polypeptide chains, they get joined by alkyl groups, and alkylation, because of alkylation, two strands of DNA, they are not separated during replication, and it leads to mutation. And another type of, last type of chemical mutagens are the acridine dyes or the organic dyes. For example, proflavin, it acts as organic dye or acridine dye. These dyes, they enter in the DNA chain between two nitrogen base pairs, and they cause addition or deletion. For example, there is a chain, the sequence of nitrogenous base is ATC, say GGC, TTT, and the dye, the acridine dye, it appears between this T and C, so it becomes AT. Now we know the three nitrogenous bases, they form a codon, it will become CGG, and CTT. And we can see this last T, this is separated, and here A and T, they are separated. The whole sequence of nitrogenous bases in polypeptide chain is changed, and this leads to mutations.